So what I was saying was the lecture is kind of broken into two parts. There's a blood part, and then there is a cardiovascular system part, which is mostly heart with a little bit of blood vessels involved. So the first part is blood. So remember that blood is a connective tissue. What is the ground substance in blood?
Plasma?
Plasma. Basically water. Plasma is just fancy water. Blood has three main functions. Number one, it transports things around our body. It's the highway that allows things to move through our body. Oxygen from our lungs to everywhere else in our body. Carbon dioxide from everywhere else in our body to our lungs. The nutrients, we eat them, they get into our digestive system, and they end up in the bloodstream, and they move throughout our body. Our hormones, by definition, are secreted into the bloodstream, so they move through the bloodstream to get everywhere. The heat, how and why are we transporting heat through the bloodstream?
What do you mean?
Why? So we're transporting heat. What do you mean by we're transporting heat? From where to where?
From your pores, like your limbs?
Perfect. Yeah. Our limbs, our fingers and stuff, are colder than our core temperature. That's why you can't just hold the thermometer in your hand and get an accurate reading. Because the further away you get, the colder it's going to get. But if we didn't have that high core temperature, with that heating up the blood, then gets pumped out here, our hands would get very, very, very cold. It's like a radiator, where you have a furnace that heats the liquid in the core and then gets pumped out to the outside. And the waste also gets transported through the bloodstream. So cells, remember, are constantly making waste. It gets excreted out into the extracellular fluid, which then slowly makes its way into the bloodstream. Once it's there, it'll go through until it gets to the liver or the kidney, depending on what kind of waste it is, and then they will take it out of the bloodstream. Blood regulates body temperature, like we talked about. It also regulates pH. How does our blood regulate pH?
Buffers?
Buffers, yes. We talked about buffers in the blood in our chemistry lecture. Do you remember what kind of buffer was in the blood? Start with a B.
B?
Start with a B, and then an I.
What was the question?
And then a C. So can you write it on the board?
Yes, please.
Yeah. B, I, C.
Okay.
Then an A.
Violet? No. I don't know. There's no E.
Who? Crystal, did you say it?
Oh, no.
No. Did you?
I said something entirely different.
Oh, well, it certainly sounded like what?
Oh.
Am I right?
Lactose?
The Cardi?
No,
The
Bicarbonate?
Bicarbonate.
Oh, wow.
Is that not what you said?
No, I said something very bad.
Okay. Well, it sounded like bicarbonate, and I have no idea what it could have meant.
I said the Cardi.
You're about to.
But we have a bicarbonate buffer in our blood, it's going to regulate pH. It's also going to regulate the water content of our cells. Our cells are kind of like sponges. They soak up water if it's available to them. They won't, just like a sponge, you can't put an infinite amount of water into them. They have a certain capacity they can hold, and then they'll stop holding more. But if you put a sponge in a dry area, it dries up. And so if you don't have water getting through your bloodstream to the area around a cell, the cell is going to shrivel up and die. And so this is going to regulate how much water gets to the cells. The blood also has some protective functions. It prevents you from losing blood. That's kind of a like a circular argument, but parts of the blood will help you clot if you were wounded to prevent the other parts of your blood from escaping. There are parts in your blood which serve no purpose other than to clot. And so when they clot, they're going to prevent the red blood cells, the parts that carry the oxygen, from escaping. It's also, remember that the immune system, those white blood cells, are in blood. And so that's certainly a protective function, the immune system. If you take a blood sample from someone, what you're getting is called whole blood. It is roughly 38 degrees Celsius, 100.4 degrees Fahrenheit. Those numbers seem a little bit off, probably, to what you had in mind. What are the numbers you normally have in mind?
98.6.
98.6. In Celsius, in science, because we use Celsius, we use 37. Blood temperature is slightly different because if you're taking the temperature of a person, you're not taking it in the absolute core. You're taking it somewhere towards the outside. And so it's not quite 100.4 degrees Fahrenheit. The pH, we said, normally is between 7.35 and 7.45. We said if you're below that, you have a condition called what? Acidosis. Remember, low pH is acid. And so if you're too acidic, you have acidosis, or we say that you are acidotic. If you're above 7.45, you're over here, you have what? What's the opposite of acid? Base. So you're basic. The real term is alkaline or alkalytic. Alkaline and basic are the same thing. In an average adult male, we have about 4 to 5 liters of blood. It all depends on how big you are. A bigger person is going to have more blood, a smaller person will have less. In that whole blood, there are 2 main parts. There's the plasma, and then there are the elements. The blood plasma is water, and then things like sugar and sodium chloride and gases and oxygen, carbon dioxide dissolved in that water. It is just a water solution. It does not have large particles in it. The large particles are these formed elements. They're going to be cells and cell fragments, which are pieces of cells. And so if you take whole blood, all the little bits are going to be formed elements. If you take those bits out, you're just going to have basically water with stuff dissolved in it, and that is our plasma. The process of making blood cells is called hemopoiesis. Hemo refers to blood. In heme, we'll talk about heme. The heme is what makes red blood cells red. It's a protein that binds to iron, and iron carries oxygen. And so heme is what makes red blood cells red, and poiesis is genesis or making of something. This happens in red bone marrow. Remember, we had 2 types of bone marrow, red and yellow. Only the red bone marrow actually makes blood cells, and these blood cells are made from stem cells. Pluripotent means that they are stem cells that can turn into multiple different types of cells. There are stem cells that can only turn into 1 or 2 different things. Pluripotent stem cells are, well, just what I said, able to turn into multiple things. I couldn't come up with a different way to say it, because the first way was so perfect. So if you have blood drawn, probably the first thing they do when they take it from you, they take it in the back room, is they spin it. You put it in a centrifuge, and so a centrifuge is going to spin it really fast. And when you do that, it separates. When you spin something in a centrifuge, it makes heavy things go to the bottom. And so if you spin whole blood, you get the formed elements down here, and then you have the plasma up here. So the blood plasma is about 55% of the volume of your whole blood. So an average blood sample will be 55% water. Under that, we have this. And on the top is what's called the buffy coat. Buffy coat is white blood cells and then platelets. I'm sure you know white blood cells are immune system cells. We'll talk about platelets, but platelets are that clotting system. And then down here, we have red blood cells, which make up about 45%. What do you notice about these two numbers?
They're a lot bigger.
They add up to 100. So what does that tell you about this? It exists.
It's a very small.
It's just very small. It's just very small. So that when you round the whole percents, it disappears. And so this is not to scale. The buffy coat is not that big compared to the red blood cells down there.
So you would be able to see the.
You can see it. It's not a thick layer though. It's just kind of like a little cap on it. It's like just a very dusting of snow on top.
Okay.
It's been a while since you looked at the blood slide over there, but if you can picture it, there were a lot of red blood cells and like 2 white blood cells. There's just a lot more of these. So we said whole blood is made up of blood plasma and formed elements. Blood plasma has things in it. It's mostly water, but we have proteins and then we have salts. You don't need to know these things. It's more for your information. And we'll talk more about these things, the white blood cells, when we talk about the immune system. That's its own lecture. But in the formed elements, we have red blood cells, the white blood cells, which are these, and then the platelets. So you can see the red blood cells, there is 4.8, so roughly 5 million of them, whereas the white blood cells, you have 5 to 10,000.
Who doesn't know those numbers?
No, definitely not. I would know maybe 55 and 45. Well, let's say those numbers you need to know. These definitely not. Unless you're looking at my blood results and you're my nurse or whatever. Then I hope you know those. But not in this class. So the formed elements we said include the red blood cells. We normally abbreviate red blood cells, RBCs. This is a lot quicker to say than red blood cells, even though it's, I guess, the same number of syllables. It's quicker to type though, definitely quicker to type. We have RBCs. They're also called erythrocytes. The white blood cells are WBCs or leukocytes. And like we said, there are multiple types of these. And we'll talk about those in detail in that lecture. I'm not going to ask you any questions about these having to do with this lecture. And then the platelets, there's not very many of those either. This is just showing that pluripotent stem cell. And again, this is more detail than you need. But you do need to just get in your head that pluripotent stem cells have the ability to make a lot of different cells. And that most of these blood cells are going to come from pluripotent stem cells. So you have one population of cells that are going to change and make all of these different cells that have very, very, very different functions. So yes, these are all white blood cells. And so they're similar to each other. But that pluripotent stem cell made white blood cells, and they made red blood cells. So here's a picture of a blood smear. And so like I was saying, there's a whole bunch of red blood cells. There are very few white blood cells. These little purple dots are platelets. And they're small because they're pieces of cells. They were a larger cell that broke apart into pieces. And so they're just little fragments floating around. The red blood cells are not shaped like a normal cell. They look almost like a donut that didn't get punched all the way through. It's a flat, rounded disc with a dimple on one side. They do not have nuclei. Why not? Cells have nuclei, right? So these are cells. Why do they not have nuclei? What's the function of the red blood cell?
Transport.
Transport. And so it's like a semi-truck. You're going to put stuff in there and then transport it. If the purpose is to use that truck to transport something, you want to fit as much in as possible. So that you make as few trips as possible. And so a nucleus is pretty big. So if you had a nucleus, it would take up a good part of that cell. So it wouldn't be able to carry as much oxygen. And so once the cell forms, it gets rid of its nucleus. It then can't divide. The purpose of the nucleus is to divide and also has the genes to make all the proteins. And so once this red blood cell gets rid of its nucleus, it is on its way out the door. It can't make any more proteins. It can't replicate itself. It has a limited lifespan, and then it dies. The pluripotent stem cells, the top of that tree, stay pluripotent stem cells. So when they divide, you get another pluripotent stem cell, and then something that turns into a blood cell. So every time it divides, you make one new cell plus a new pluripotent stem cell to take the place of the one that just divided. Red blood cells live about 120 days. And so, does anybody ever give blood? You remember how often they say you can do it?
I think it's like 4 months.
How many days are in 4 months?
120.
120. So if you give blood, they won't let you give blood again until your body's had a chance to completely turn over the blood cells in your body. And so if I went and gave blood today, I would lose some when I donated. But in the next 4 months, they would have died anyways and been replaced. And so after 4 months later, it's right back where I started. Erythropoiesis is the formation of red blood cells specifically. Hemopoiesis is the formation of blood cells. Erythropoiesis, referring to erythrocytes, which was another name for the red blood cells, is the formation of red blood cells in our red bone marrow. If we become hypoxic, what does that mean? What is hypoxia? Not enough oxygen. Oxygen, hypo. Like hypothermia. Not enough oxygen, hypoxia. If we don't have enough oxygen, we are going to stimulate red blood cell formation. Why? Why would we want to do that? Yeah. If we don't have enough oxygen, we need to get more oxygen in our body. And so, most likely in that situation, the red blood cells that we have are maxed out. They can't carry enough oxygen. And so we need to get more of them. the kidneys can actually sense oxygen levels. And so if oxygen levels go down, the kidneys will recognize it. The kidneys will secrete something called erythropoietin. That is going to stimulate red bone marrow to make red blood cells. Makes sense? You're measuring if you have enough oxygen. If you don't, wherever those sensors are, which happen to be in the kidneys, will send a signal in the form of erythropoietin to the red blood cells, to the red bone marrow, the factory, to make more red blood cells. Red blood cells, though, we said, only live for 120 days. We have a lot of them. We have a lot of red blood cells. And so if every time a red blood cell died, they got jumped, we would lose a lot of nutrients. Because a lot of nutrients, a lot of energy go into making a red blood cell. And if that many of them are going to die every single day, complete turnover every 4 months, we need to recycle a lot of those parts. So here we have blood that has been in circulation for 120 days. It's going to die. We have a red blood cell that is going to die. So here it dies, and we break it into parts. The most important part of that red blood cell is the hemoglobin. The hemoglobin is a very, very, very large protein that carries the oxygen. Plasma membranes are easy to make again, but hemoglobin takes a lot of energy, so we don't want to waste it. The hemoglobin is going to get broken down into its two parts, heme and globin. Globin is just a standard protein. There's nothing really special about it, but other than it's big. And so we just take that globin, we break it down into amino acids that can just be used again to make another protein. The heme, though, has iron in it. And iron is what actually carries the oxygen. Hemoglobin carries iron, which carries the oxygen. We don't have a ton of iron in our diet. Iron in a normal diet is not rich. And so we cannot continuously lose that iron. And so the iron from the heme gets saved by a protein called transferrin, but you don't need to know that protein name. You need to know the iron gets saved, though. And then it gets transported to this. What's that a picture of, and why is it going there?
Femur.
It's a bone.
So they stole
It's a
it.
femur, yes? Red bone marrow. In the, what's this part of the bone? Epiphysis. The red bone marrow is in here, and so we're going to take the iron, and we're going to take it back to the factory to let the factory reuse that iron. The protein part of heme gets converted from one thing to another. Biliverdin is a middle ground between the heme and bilirubin. You might have heard of bilirubin before. Where have you heard of bilirubin? Anybody? A lot of times when children are born, their livers aren't fully functioning yet. The liver takes bilirubin and digests it. Bilirubin is yellow. And so a lot of very young, just newborns, will get what's called jaundice. Jaundice is where a person literally turns yellow. If you're light-skinned, it's very easy to see. In everybody, it's very easy to see in the whites of your eyes. The whites of the eyes are made of what? What's that part of the eye called? Sclera. And so, if you see someone with yellow eyes, that generally means they have jaundice, and that means they probably have liver failure. Could be for a lot of different reasons. In a newborn, it's most likely just that the liver hasn't kicked into gear fully yet. But Luckily, bilirubin is not stable in sunlight. And so if you put the little kid under a sun lamp, it breaks down the bilirubin and you basically bypass the liver. And so if they're in the hospital, they have sun lamps. If they want to send the kid home, but the liver's still not really up to par, they'll actually give you a sun lamp to take home. If you just have just a little bit of jaundice, they will literally tell you, set the kid in the window. That's a real thing. You just put the kid in the window and he changes color.
Person.
Yes.
Or
Or she. Yes. Bilirubin, when you break it down, turns into a brown color. And then it works its way through all of this, through the small intestine, the kidney, and some of it goes into the intestine, some of it goes into the kidney. Now why do you think brown color is important? Where's some of it going? Into the intestine. What's brown in the intestine? I think you all know, right? That is why feces are brown. It's the bilirubin that started as hemoglobin. The brown in feces is the leftover digested part of red blood cells. Okay, so then there are the white blood cells, and again, there's a lot of names here. We'll come back to these names. I'm not focused on the white blood cells today. Focus on the red blood cells and the platelets. The white blood cells have nuclei. So if somebody's going to test your DNA, they can take a blood sample. But in reality, they're only testing your white blood cells. Because the red blood cells and platelets don't have any DNA. But luckily, we don't need very much DNA to test. So if you take a sample, even though there's very few white blood cells in there, that is enough. The white blood cells can be granular or agranular, and there are multiple types, but the granular and agranular just describe what they look like. Granular leukocytes have little specks in them. Agranular do not. The white blood cells fight inflammation and infection. Primarily the infection part. The inflammation is an incredibly complicated thing. Neutrophils are going to attack bacteria. Macrophages attack things by eating them. Macrophages are big. They use phagocytosis. We've seen macrophages before. Where did we see macrophages? Yes. In the diagram of the skin, we had this big spider-looking cell. It's an intraepidermal macrophage. They're big cells. They literally go around looking for an invader, and when they find one, they just swallow it up. That is phagocytosis. The eosinophils are related to that inflammation. And so if you take blood work and you see high eosinophils, that probably means you have inflammation somewhere in your body because it's trying to fight that inflammation. Basophils are also involved in the allergic response. Lymphocytes, there are 3 types of lymphocytes: B cells, T cells, natural killer cells. Natural killer cells definitely have the best name out of all of them. The B cells make antibodies. B cells make antibodies. That's literally how I remember it. It doesn't really make sense. It's not the best mechanism, but it works for me. So B cells make antibodies. T cells attack things other than bacteria. They don't attack bacteria, they attack everything else. Whether that be viruses, fungi, we don't have mushrooms growing in it. What is?
This one.
We get fungus infections, primarily on the outside of us, but we can get internal fungal infections in cancer cells. Our body is capable of recognizing a cancer cell the instant it forms. And it goes, and the immune system kills it. As we're sitting here, we are constantly making cancer cells. But as soon as they're made, they get killed. And so in order to actually get a cancer, a tumor has to avoid the immune system. They have to avoid putting up that flag saying, I'm a cancer cell. And there's a whole lot of other things that have to happen. And so there's a very, 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 very tiny chance that when a tumor cell is made, it actually turns into a tumor. If there was a high chance, we would all have tumors and be dead by 6 months of age. It's bad. But the immune system does a very, very good job of doing that. One of the newest ways of trying to fight cancer is, rather than trying to kill the tumor itself, teaching the immune system to go in and kill it. So the only thing stopping that the immune system from killing a tumor is that the immune system doesn't realize that's a tumor. There's something about it the immune system doesn't recognize as a tumor. And so if you either plant the flag, the tumor flag, in the tumor, and so then you don't have to teach the immune system to do anything, then it's just going to do what it naturally does, or you can actually teach the immune system, this is a tumor. I know it doesn't have a flag, but kill it anyways. And so that's actually turning out to be a fairly good way of going at it. White blood cells do not live very long at all. Red blood cells last 4 months. White blood cells, hours or days. Why do you think that is?
Because they're constantly fighting out.
They're constantly fighting things. Yeah. Red blood cells, they just go about their business. And all they have to do is float around. They literally don't do anything except float in the stream. White blood cells are out there fighting with things. And so they're constantly under attack. So they're not going to live very long because they're constantly under attack. And also, what is invading us is constantly changing. So we're going to learn that some of these white blood cells are specific for certain invaders. And so we can't maintain a large population of cells for every single possible invader. We're just not capable of it. And so when we get an invasion, we make a bunch of cells to fight that invasion, but then once that infection is gone, we don't need those anymore. And so we stop making them. So once they die, we just let them go down. Then we get another invasion, so we make these different cells to fight that thing. The platelets, we said, are fragments. Under the microscope, they look like cells. It's crazy. They're shaped like a cell, but they're actually a piece of a cell. These do not have nuclei, and they came from something called a mega karyocyte. Mega meaning very big. So a megakaryocyte breaks apart, like just shatters, and you get a whole bunch of little fragments out of it. Those fragments, the platelets, make what's called a platelet plug. And so if you have a damaged blood vessel, you're bleeding, there's going to be two processes that happen here. The primary clot is going to be a platelet plug. The only purpose of these platelets is to block bleeding. Hemostasis is that process of stopping bleeding. Okay? When a blood vessel is injured, we have a gap. It's like a cut or a hole in a hose. The water, the blood starts leaking out. We can stop that blood loss 3 different ways. The one we just talked about was platelet plug. That's going to be the long-term solution. We can also, in the short term, undergo what's called a vascular spasm. Based on the word, what does that sound like is going to happen?
Constriction of the blood vessel.
Yeah. If you have a blood vessel that is leaking, you just constrict that blood vessel to limit how much blood is actually going into that blood vessel. If you cut off the flow or reduce the flow, you're going to lose less blood. The hole is still there, but there's not much blood getting out of it. And what you probably think of is just coagulation. This is blood clotting. Blood clotting is the other parts of the blood, primarily the red blood cells, getting stuck together. Red blood cells are big, at least compared to platelets. They're very big. And so they make a pretty good plug. Quick plug, though. Imagine, think if you have pebbles, okay? If you have pebbles and you pour them into a jar, you can imagine how much empty space is in that jar, right? If you have small rocks, and you fill the same jar with small rocks, what can you tell me about the amount of empty space in that jar?
Very small.
There's going to be
More.
more. If you have big rocks,
Do we have jar?
if you have just a couple big rocks, 3 big rocks, there's a lot of empty space. If you have pebbles, they can fit in there a lot tighter. What if you put sand in there?
No space.
There's going to be basically no empty space. So smaller things packed together better. So which do you think is better at preventing blood loss?
Like,
The very small ones. They would be like the sand. They pack together very tightly and make a very tight seal. Red blood cells would be more like the pebbles. There's a lot of them, they're big, and so you don't need very many of them to block most of the blood loss. But they're not the long-term solution because the plasma can still slowly get through them. Another thing we talk about in terms of blood are blood types. And most people have no idea what that means. They might know what their blood type is, but they have no idea what that tells you about them. If you look at a red blood cell, on the surface of that blood cell are things called antigens. An antigen is a generic term for something recognized by an antibody. And so antibodies are proteins that stick to an antigen that they recognize. And every antibody is designed to stick to a very, very, very specific antigen. If you change that antigen, which is usually a protein, just a little bit, that antibody doesn't recognize it. And so we have a red blood cell. On the surface of that red blood cell are antigens. And they're basically those little flags like a tumor has. So we have a red blood cell, and then there are antigens, little proteins, stuck into the membrane, but then hanging outside of the cell. They're mostly glycoproteins, which are proteins with sugars on them. There's also some glycolipids, which are a combination of sugar and lipid. And so these little antigens are going to tell the immune system and the other cells around that red blood cell what type of cell it is. When you figure out what kind of blood type you are, all it's saying is what type of antigen your red blood cells have on them. And so there is A, B, and O. There are 2 antigens, A and B. Okay? There is no O antigen. And we'll see where the O comes in now. If you look at your blood type, you're going to be classified, you could be classified as RH. There is a different type of antigen. There's 2 types. So we have the squiggles and we have the crosses, let's say. Some people have the crosses, some people don't. And so everybody at least most everybody has V's, but let's say half the people have it. So you're going to have A, B, O, or there's going to be a positive or a negative. Like there's A positive or O negative, things like that, right? The letter responds to these antigens. The positive negative responds to these antigens, which are the RH antigens. So we don't say RH, we just say positive or negative. And people who know what they're talking about know what they're talking about. So this is the A, B, and the O. And so if you are type A, that means that you have the A antigen on your red blood cells. If you are blood type B, that means that you have the B antigen. If you are AB, you have both. Where it starts to get complicated and why it matters what blood type you are when you get a blood transfusion is if you are type A, and your red blood cell has the A antigen, in your bloodstream are antibodies that recognize the B antigen. And if you put a red blood cell into your body that has the antigen that your antibodies recognize, your immune system is going to attack it. And so if you are blood type A and you get a blood transfusion from someone that is blood type B, you just put a whole bunch of things into your body that your immune system is going to kill. And so number one, you're going to kill all the red blood cells you just got. So that's not very helpful. But also, your body, which is in very bad shape, otherwise you wouldn't have been getting a blood transfusion, is now focused on killing all this stuff. And so it's going to make you in even worse shape than you were to begin with. And you may actually put you into anaphylactic shock. You have an allergic reaction, your throat closes up, and you die. So that's why you need to know what blood type you are. If you are AB, you have neither antibody. Let's think about this. If you are A, why do you have a B? Well, let's turn it around. If you are type A, why don't you have antibody to A? Because then you would kill yourself. Same thing with B. If you're B, you can't have an antibody to B, otherwise it just wouldn't work. If you are AB and you have both, you can't have either, right? If you had either antibody, it would attack your red blood cell. If you are O, you don't have any antigen, which means you have both antibodies. And so, what do you think is more useful? Blood from someone who is AB or someone who is type O?
Depends on who you're doing.
AB.
If you're type O, you can only receive O, but if you're AB, you can receive from anybody.
Right. Okay. So, if you are type O, if you get blood from A, B, or AB, you're going to attack it. And so you have to have O blood. If you are A and you receive O, will it work? No, it won't. O's are only interchangeable with each other. If you are O, you can only receive from an O, and if you are O, you can only give to an O. What if I am AB and I don't donate my blood? Who can they give my blood to? A, B, AB, or actually they can go to O also. There are no antibodies there. You have the most useful blood in the world. It can go to anybody. So I'm not AB, and so Life South is not constantly hounding me. I would imagine if anybody is AB, they're probably calling you every 4 months.
And O.
And O, yeah. But if you're one of the majority of the population is either A or B.
Isn't O like the least like common?
Between A, B, and O, I don't know which one more. But they're definitely less common than A and B. Let's take a break. We'll come back at quarter after on that clock. got a lot of slides to get through, so we're going to go. So we're moving on from blood, and we're going to start on the heart. If you're looking at the slides, no, we're not getting through them all today. We're not going to rush through 45 slides in the next half hour. So we're going to talk about the heart. And so what you're looking at here is a diagram of the walls of the heart. Probably the very first lecture, we learned about the different membranes. We had a membrane around the heart. What was that called? You remember? Pericardium. And so those membranes had an inside and outside membrane. Outside was the parietal, inside was the visceral. So what you're looking at here is the wall of the heart with that membrane. And so this is the pericardial cavity. This is just membrane out here, and this is heart. And so they've labeled this part here as visceral pericardium, which is also called the epicardium. So the visceral pericardium is literally the outside of the heart. It's not just a membrane around the heart, it is the outside layer of the heart. And so the heart itself has three layers. There's the visceral pericardium and the epicardium. There's the myocardium, which is large. And then on the very inside, there's a very thin layer called the endocardium. Endo is in, right? The innermost layer is the endocardium. Myo, think middle. And then the epi. Epi is outer or upper, which is also the visceral pericardium. So when we look at the heart has four chambers. There are four distinct open areas in the heart that are capable of closing off from each other. Those are the chambers. The two on top, there's two on top, two on the bottom. The two on top are called atria. Think of the atrium of a building. It's where you first go into. Okay? It's just inside the door. And so blood is going to enter the heart in the atrium. The lower two chambers are called ventricles. The ventricles are going to be larger, and they're what's going to really propel blood out of the heart into the rest of the body. We have the atria on top, ventricles on the bottom. Between the two atria, between the left and the right atrium, is the interatrial septum. A septum is a thin wall that separates two things. So this is a septum between the atria. So it's the interatrial septum. It can also be abbreviated IAS. Between the ventricles is the IVS, the interventricular septum. So here we see that. So we have four chambers. 1, 2, 3, 4. The top two are the atria. Between them is the interatrial septum. And then down here are the ventricles with the IVS, interventricular septum. So immediately we can see the atria are attached to big blood vessels, and the ventricles are also. So you can't just go by that, because some of these are bringing blood in, some are taking blood out. When you look at a diagram like this, for the most part, things are going to be oxygenated and or moving away from the heart. Blue is going to be deoxygenated, more or less coming back to the heart. Blood leaves the heart to go to the rest of the body after it has oxygen. It goes to my foot, and then it comes back. When on its way back, it has used up its oxygen. It's just coming back to the heart to get more oxygen. Now, there are two ventricles because we need to pump blood two different places. Where do you think those two different places are? The lungs in the body, yes. Unoxygenated blood has to go to the lungs to get oxygen. And so we're going to pump some blood to the lungs, and then it's going to go to the lungs, get oxygenated, come back to the heart, and then get sent off to the rest of the body. So this atrium here is receiving blood. It's an atrium, and so it is receiving blood. That one, which is the left atrium, because it's anatomical position, on your right is the patient's left. It is receiving blood from the heart. From the lungs. It is receiving blood from the lungs. If it's receiving blood from the lungs, it is oxygenated. And so it's red. It comes in here. It's going to then pump it down into the left ventricle. It's still oxygenated, so it's red. And then it is going to get pumped out the aorta into the rest of the body. It's going to go into the rest of the body, do its thing, and then come back. When it comes back, it's deoxygenated, and so it is blue. We have blood going to some places above our heart and some places that are below our heart. And so it's coming in from two different directions, down below and up top. But they both dump in to the same place, the right atrium. It comes going to come in here, get pumped down into the right what? Ventricle. It's going to get pumped from the right atrium down into the right ventricle, and then it's going to go from here to where do you think? The lungs. Yeah. You have the deoxygenated blood. It's not useful until it's oxygenated. And so once it comes back to the heart, it gets pumped to the lungs. Comes back from the lungs, now it's ready to go, so we're going to pump it out to the body. But then it's deoxygenated, so it goes to the lungs, heart, body, heart, lungs, back and forth. There are also valves in that heart. There's some pretty high pressure going on in the heart and the blood vessels around the heart. And so if we don't have valves, the blood's just going to go both directions. But we need blood to continue in a certain pattern, otherwise it doesn't work. If we have deoxygenated blood going backwards to where it just came from, that's going to prevent the oxygenated blood from getting there. And we don't need oxygenated blood going back to the lungs. We just don't need it. And so we're going to have valves. And these valves are one-way valves. They're going to allow the blood to flow in the normal direction, but if you try to go backwards, it's not going to work. Imagine a door with a little push bar on it. If you walk up to the door and you're going to push it the way it's meant to go, it's going to open. But if you walk up to a door, the same door from the other side, where there's a handle, and you push, nothing's going to happen, right? The door only swings one way. It's designed to get pushed open one way. So these valves are going to do that. And they're going to open and close at certain times. They're going to open when blood needs to flow one way. But once it stops flowing, it's going to shut to prevent what just went through from coming back. The atrioventricular valves, or the AV valves, separate the atria and the ventricles. Atrioventricular. They're just the valve the blood's going to go through to get from the atria at the top to the ventricle on the bottom. And then there are semilunar valves which sit in the arteries where the blood is leaving the heart. And so once you pump blood out of the heart, these semilunar valves prevent it from backing up into the heart. So here we have our four chambers again. And so between that atrium and that ventricle, and then this ventricle and that atrium, we have two different atrioventricular valves. And they have their own names. They are the bicuspid valve and the tricuspid valve. Bicuspid is on the left side, the tricuspid is on the right side. Then we have two of these semilunar valves, the aortic and the pulmonary. Luckily, these names describe where they're going. The pulmonary semilunar valve is on the artery that is going to the lungs. Pulmonary is lungs. And the aortic semilunar valve is in the aorta. And the aorta is the very, very, very large blood vessel that carries all of the blood from the heart and then branches and goes to different parts of our body. So there are four valves. These four. Remember that the whole thing is based off of pressure. It's all based off pressure, blood pressure. In anything, whether it's blood or air or water or anything, is going to move from high pressure to low pressure. So when the heart is not contracting, you have more or less the same pressure throughout your system. But when it contracts, all of a sudden we have very high pressure in the heart. It's higher than in the blood vessel, let's say the aorta, leaving the heart. And so it'll go from the high pressure in the heart to the low pressure in the aorta. So that's what's going to push it from the heart into the aorta. So we can control the blood flow, not using that to our advantage. We know blood is only going to flow when we create areas of high pressure. And so we can control that by contraction and relaxation of the heart. In the part of the heart that actually contracts, where that cardiac muscle is in the myocardium. In that first slide we showed where there were the three layers of the heart. The myocardium was the middle one, M for middle, and it was by far the largest one. Most of the heart is cardiac muscle. There's a lot of muscle in there. And we can also control the movement of by using the valves. Even if we have a split second where the pressure reverses. Because when the heart expands, all of a sudden the heart is a low pressure area. And so we don't want to suck back in the blood that we just pumped out, which is where the semilunar valves come in. And so even though the blood wants to flow from high to low pressure, in that instant it wants to go into the heart from the aorta, it can't, because the valve is blocking the way. Here we see the valves. So we have blood coming in from the lungs. There's a valve here. It will only open when the atrium contracts, creating high pressure to push it through that valve down into the ventricle. Then, when we pump it out of here, there is a valve in the aorta. So we pump it through, and then when the heart expands again, it can't get sucked back in. The same thing is going on over here with the blood going between the right aorta and ventricle and then going over to the lungs. Just remember the valves are one-way things. They always keep blood flowing in the right direction. That's what we just talked about, but it's just a good summary for you. You need to know, at this point you need to know this part. We'll come back to the lower part. But if you know the flow in the heart, including the lungs and the rest of the body, that's what you need to know. So this contraction, the contraction of the heart is controlled by electrical signals. And in our heart is something called the sinoatrial node, or the SA node. This is a term that people generally actually do use the acronym. People use the term SA node a lot. This is in the right atrial wall. So it's in the right atrium. And this is what starts the cardiac contraction. This is your pacemaker. we naturally have pacemaker in our heart. They're cells that at a certain impulse time rate will send out an electrical signal. They just do it. And their control, how often that is controlled by your autonomic nervous system. They're telling the SA node how often send that electrical signal. When they send that electrical signal, it's going to send it to the atrioventricular node. And then after the atrioventricular node, which is the AV node, the action potential is going to slow. So this SA node, its purpose is to start the process. It doesn't actually cause a contraction. It just is the timekeeper. It then sends that signal to the AV node. The AV node is in the interatrial septum, which is the wall between the two atria. Once the signal gets here, it's going to amplify the signal. The AV node is very good at making a strong signal, but it can't keep time on its own. The SA node keeps time for the AV node. The AV node is going to cause the atria to contract. So the top two chambers of the heart are going to contract before the bottom two. And it makes sense because the purpose of the top ones is to pump blood into the bottom ones. And the purpose of the bottom ones is to pump it elsewhere. And so if the bottom ones pump before the first ones, there's nothing in the ventricles to pump. So we need to pump the atria first to get the blood down into the bottom, and then the ventricles will pump it out. And so we need to slow everything down at that point. We hit the AV node, the atria contract, but it also puts a hold on the whole process. Because the atria needs time to contract. It is not an instantaneous process. It is a large muscle that has to act. So the action potential is going to slow down. So here we have the SA node, and we have the AV node here. So the SA node is going to send the timekeeping signal to the AV node, which is going to make the top two chambers contract. After it finishes contracting, the action potential is going to continue. The AV node slows it down, but it doesn't stop it. And so then it will then continue down into the ventricles, so they'll pump. So SA node to the AV node. When you get to the AV node, the atria contract, and then the ventricles contract. We can measure this happening. I think a lot of people have either had or given an EKG or an ECG. So we can measure this action potential going through a heart. It's not, that electricity is not contained solely within the heart. So we can just put sensors on a chest and actually measure the electrical changes going on in the heart. That's called an electrocardiogram. And so it's ECG, even though most people think of it as an EKG. The K is from German. Germans spell cardio with a K. And so it was initially EKG, but in English it's actually ECG. But ECG and EKG are the same exact thing. If you're measuring that, that is where you see that heartbeat that they like to show on TV shows. It's always beeping in the background with a very nice little curve, and then just when the killer is about to tell why he did it, it goes flatline again. Right? So we're going to measure this electrical signal using our electrocardiogram, and we're going to learn how to read, at least at a basic level, what the electrocardiogram is telling us. When we do it, we're going to see three peaks. There's going to be 3 peaks. The first is called the P wave. And so we're coming along, and we're going to see a little bump. And then it's going to continue on again. This is, correct me, Mark? That's the P wave. P wave is when the atria depolarize. So depolarizing is the contraction of the atria. That's what we're seeing here. So the atria have contracted, and then just after the P wave ends, we get, it depolarizes, and there's a slight delay. And so the actual contraction is happening kind of in this area here. We got the P wave. That's the atria contracting. And then we have what's called the QRS wave, or the QRS complex. And this is the big spiky peak. So it's going to come along, and it's going to, you're going to go through this big thing. So it's going to drop a little bit, spike way up, come way down, and then come back. We're going along, we drop, we spike, we come down, we come back, and now we're back here. So this is Q, R, S. This whole process is the ventricles contracting. Atrial contraction, ventricular contraction. And then after that, we get another little one. And that last little one is the T wave. This is when the ventricles repolarize. This is an action potential. And so when this is happening, all that sodium is flooding in. Remember, after we depolarize, we have to repolarize. And there's electricity associated with that. And so we can measure that. And so we have P, Q, R, S, T. And so people who know how to read this can tell, based on the different peak heights, if something's going on. They can tell, based on how far apart they are, if something is wrong. They can tell by the shape and how far apart they are, and they can read all kinds of stuff into that I have read. I know if it's flat or a peak is missing, go to the hospital. That's what I know. So when we look at this process, that whole cardiac cycle, we break it down into 3 parts. There's the relaxation period where nothing is contracted. Diastole is the absence of contraction. So it's as big as it's going to get. Systole is contraction. And so we have diastole, where neither is contracted. And then we have atrial systole, where the atria have contracted. And then we have ventricular systole, which is when the ventricles contract. Here's our relaxation. Relaxation, so everything's just sitting there waiting. Then the atria are going to contract, atrial systole, and then ventricular systole to pump everything out of the heart. We can also hear this. The lub-dub that you hear is this going on. What you're actually hearing is not the contraction. It's the valves. The valves are noisy valves. So when the valve closes, you hear it. Just like when the door slams shut, you can hear it. You can't hear a door open unless you have squeaky hinges. But if somebody slams the door, it's going to make a big boom. And so the first sound, the lub, is the AV valves closing. And so the atria contracted, they pump the blood down into the ventricles. But before the ventricles contract, we have to close the valves to prevent it from going back up into the atria. So that's the lub. The second sound, which I think everyone except the person who wrote this book considers a dub, is the semilunar valves closing. That's after the ventricles contract. We push the blood out of the heart. The semilunar valves are going to close, prevent it from coming back into the heart. They say it's a dump. I've never listened to a heart with a stethoscope, but since you were little, they taught you, Big Bird always taught you it was a dump. Right? So it's lub dub or lub dub. But lub dub is easier to say. Cardiac output is just a measure of how much blood your heart is pumping. And so how much blood your heart is pumping is only based on two things. How big your heart is and how fast it's pumping. And we can't control how big our heart is. And so if our body wants to control how much blood our heart is putting out, it controls the rate. And we learned that comes from the medulla, whatever, oblongata. Heart disease is still the largest killer in the US, but not for long. Cancer is going to overtake it. That's good and that's bad. That's not because cancer is getting more prevalent. It means that we've done a good job of trying to prevent heart disease from killing people. So heart disease is coming down. It's going to go down below cancer. We've done not nearly as well with cancer, which is the bad news. It's not that cancer is spiking up. Cancer is staying more or less the same. It's just heart disease is dropping. So there are a lot of things that make someone predisposed or more likely to develop heart disease. And a lot of these things are things that you know. Age. You get older, you get heart disease. Generally, people who have heart disease are older. As we get older, things get clogged up, the heart gets weakened, we get damaged, things like that. So the heart stops working as well. Males are also more predisposed to heart disease than females. It can be familial. And so if someone in your family, particularly someone of the same sex, so if you're male and there's someone in your family that was male that had heart disease, you are at increased risk. If you're female and your mother or your grandmother or someone has heart disease, you probably have a higher risk. Smoking increases heart disease risk, as does diet. High blood pressure does also. If you have high blood pressure, it's going to make the heart work harder. It's also putting a bunch of pressure in like in a balloon. It's constantly trying to get pressure out. High cholesterol causes problems in collection in your veins and your arteries, which can cause something to break away, move through your bloodstream, and clog your heart. Inactivity, just living a sedentary lifestyle, is a risk factor for heart disease, and so is stress. Remember, we learned stress can be bad for your heart, primarily because stress is going to cause high blood pressure. So all of these things can lead to a greater chance of heart disease. This is a natural stopping spot, so we will stop. Start lab at 8.